And hello guys, before going to that, we have different alleles. One is a wild type, another one is a recessive, what we call the mutant type. Well, during this generation, during the evolution process, the damaged DNA, which is unable to repair itself, it will remain the unrepaired DNA. And on subsequent generation it passes, and whenever there's accumulation as a defective one because of this homozygote recessive, both the alleles, that leads to the defective one, and it might lead to the unusual phenotypic characters of the cell, and that might lead to the death of the cell. In case the cell survive, it might be the tumor one, or later on it might be becomes a cancer cell which is rapidly proliferate, and that's really very harmful for the cells. And in our genomes, there is a huge amount of non-coding sequence. That's mostly the junk DNA. So what we see here that the selector mutations, the junk DNA, and the coding sequences, the neutron mutations, the junk DNA, the coding sequences, the mutations. So here the defective alleles, that is because of this coding sequence mutations, that leads to this loss of alleles from this gene will be as this species unable to survive. In the coding sequence, the mutation leads to the formation or what we call retention in the gene pool, because the coding sequence is not affected. So in many ways, and also one more thing is that like your PEG6 and other very important genes remains very conserved, and the chances of getting mutation is very rare in this case also. Anything else? Let me check this out. Well, in this region you can see here with the mutations, it leads to a different cell line production, which might be the cancerous or tumor cells if it rapidly proliferate uncontrollably. And there's here a biomes are using the green fluorescence protein. It's the what we call the vascular endothelial cells being traced, because cancer cell has a property to metastasis. That means it migrate to the another region where it can grow again as a secondary tumor cells. RNA polymerase that gives rise to the formation of different proteins. Here the red and green colors shown as different types of cancer, and you can see here the higher than the average level of expression is indicated the red pixel. The red color is your protein that are expressed much higher. For example, you can see here this DNA is expressed as its protein components. So this protein formation from this mRNA is higher in those region which is shown red in color, but the opposite is in the green color. In both the cases, whether it's overexpressed or lower expressed, that leads to the cancer or any kind of tumor cells growth. Okay, so here is the important thing that how the genes being controlled. Well, there's two control region in this DNA. One is the enhancer, another one is the promoter region. The promoter region has a data box which is required for the binding, helps to bind those transcription factors to specific to that promoter region, and the enhancer on binding with the activator. Remember that the enhancer and the promoter both are nothing but the segment of DNA having some specific sequence which is conserved in nature. So the activators on binding with those enhancer, and after that, with the help of some mediator protein, it binds with the help of your transcription factor. This looks like something this. So you can see this mediator protein that proper in color bind with those of your transcription factor and your activator that binds with the enhancer and your promoter. So, and with the help of DNA bending protein, which helps to bend these DNA segments to close approximation of those promoter with the enhancer. The RNA polymerase, with the help of your transcription factor, that binds with your promoter region, and then the synthesis of protein, RNA, is ours. In this way, it's controlled. So if there is any kind of defect in this region, it's gonna lead to cancer cells. And then here we can see histone protein also has some important role. For example, the H1 histone is a linker, not itself. The S3, H4, H2, H2B, this histone protein unbinding, this DNA molecules not allow for transcription. So any defects there can be cause cancer. Other is your CpG acetylene with methylated. This methylated region don't allow for transcription, so any kind of mutation will be problematic. Similarly, those microRNA, this inhibit this expression of this mRNA molecules. So any kind of mutation there will also affect and lead to the cancer or tumor. So this is the thing. Thank you very much.